Hi everyone, my name is Arielle and I am a 2024 OFIS mentor, and I'm going to talk to you guys a little bit today about what I do with my research and how I spend my days as a PhD student. So, like I said, my name is Arielle and I am a PhD student here at Northeastern University in the Department of Chemical Engineering, and I work in the Materials for Tissue Engineering lab here. So what we do is we study biomaterials and their effects on cells and how specifically we can use biomaterials to help heal and regenerate the peripheral nervous system. So a little bit of background of what the peripheral nervous system is. So our nervous system is split into two parts. It is the central nervous system and the peripheral nervous system. So our central nervous system is our brain and our spine, and everything else, all of our other nerves, are the peripheral nervous system. So essentially, the information that we receive through the central nervous system through our brain then gets relayed to our peripheral nervous system, and we get sort of that physical effect. So this corresponds to our abilities to feel pain, our abilities to feel weakness, our sensory motor skills and all that. These are all peripheral nerves that dictate these things. So when we have peripheral nerve injury, this can come about through traumatic injuries. So for example, if you were to hit your hand really hard and your finger goes numb, that is an element of peripheral neuropathy or peripheral nerve injury. As well, in aging populations, peripheral nerve injuries are really common in forms of Alzheimer's or dementia, things that affect our sensory motor skills as well. And athletes, if I'm talking to any of them, you guys, we experience a lot of peripheral nerve injuries through, different tears that we experience, like ACL tears or muscle spasms. These all have elements of peripheral nerve injury that are encompassed in them. So with all that being said, in my lab, we study them at the cellular level, and we see how we can take these neural cells and encourage them to migrate faster for increased regenerative or healing effects. So I wanted to show you guys a little bit of what I study. So what we're looking at here are Schwann cells. Schwann cells are a type of neuronal cell that's found in the peripheral nervous system, and we are looking at these on a laminin substrate. So laminin is a peptide that's found in the body that's a part of our extracellular matrix that our cells like to move on. And so we have put laminin on these surfaces, and I'm tracking how they move over 24 hours. So if I were to press play, we see them kind of vibrating around, and what I do is I take each individual cell. So like, say for instance this one, well, before it died off, they start moving in different directions, and what I do is I create cell tracks of these. And in the future, what will be happening is that I'm gonna have different surface treatments, so we'll see the cells behave in a certain way. So we can visualize it a little bit. My surface treatments that I'm gonna do will be in gradient format. So I'm gonna have a gradient from low to high of a certain peptide or sequence that cells like to adhere to, and hopefully what we'll be able to see is that our cells we'll see our cells slowly moving in the direction of the higher gradient, because of course our bodies move from low to high. We go towards like what we like, right? So right now this is a constant surface that we have. That's why our cells are randomly moving. So this is what we call random migration. So if we look at, let's say, this individual cell, how it kind of like fuses with each other, there are different phenomena we can see here. Sometimes they fuse, sometimes they'll divide. This is what we call mitotic cells. So cells go through a mitosis and they kind of divide. So we're looking at individual cell paths and just their overall behavior. I want to highlight a instrument in the room. So this is our microscope where all these videos are taken in. So this is the eyepiece that we look through everything through. If we open the door here, we have our stage where I'll be putting my cell flask in, and then this is the microscope itself, so what we visualize it through. Well, actually, if you look at this is the objective lens at the bottom, so it visualizes from the bottom up. And this part is our condenser where we are able to focus the light that goes through our plate. This piece of equipment is extremely expensive and our advisor gets very upset if we were to ever break it. So yeah, this is the moneymaker here. So to tie everything together, I wanted to sort of bring everything full circle and to explain how my research affects populations. So my research, I'm specifically looking at age and sex-based differences. So how our regeneration in our body of our peripheral nervous system differs between male and female, and how as we age, essentially we need more systems to help regenerate our body, because as you can imagine, if a baby or a kid were to get hurt, they'd probably heal much faster than an older person. So this is why my research is relevant, because as we only have an aging population and our ability to want to stay young and mobile, this all comes down to our ability to heal in the body. So creating biomaterials is a good way for sort of being a therapy for a peripheral nerve injury. And yeah, hopefully that makes sense. And if you guys have any other questions about my research, I'd love for you to reach out to me. My email is efotte.a@northeastern.edu, or you could hit me up through the OFIS platforms, and I'd love to talk to you guys more about my research.